Good afternoon. So we'll try and get to the exams Wednesday. Esther's working on them. So we have one more exam, final exam. It is cumulative, but it's going to focus on the material from now on. And I will give you review questions that will tell you basically what will be on it. So everything I asked you on the previous exam was in the review questions. So the same thing will happen with the final. Okay? So I will, for the earlier lectures, we'll give you review questions that focus on what we want you to remember. Keep up with the quizzes. There will still be a quiz every week. Because if the course is over and you haven't done it, I can't reopen it for you. Okay, so try and keep up with them. There will be one extra credit opportunity for 10 points, so you can add to your grade that way. That'll be up in about a week or two. Okay? Now today and the next time, we're going to talk about two different patterns of infection. Today is acute, and it's actually appropriate that I'm doing the acute and Dr. Silverstein is doing the persistent infections, because he is persistent. And what is an acute infection? Did you burn out my batteries here? No, it was the microphone. Well, I can do it by hand. Okay, this all has to do with the pattern of infection that a virus undergoes. So you remember from what we've talked about already, there are some viruses where if you infect the cell, they will kill the cell. They will make a lot of viruses. We call those cytopathic viruses. It's quite clear. There are also some infections where there are viruses produced, but the cells are not killed, the so-called non-cytopathic infections. And we talked about how in the host, in these kinds of infections, the disease is largely immunopathological. There are also some infections, I don't think we've really talked about this, which neither killed the cell nor produced progeny. Although you could view a retrovirus infection as doing that, the retrovirus getting into a cell, integrating and not producing any virions. So these kinds of patterns of infections, lytic or non-lytic or a combination of the two, together with what your host does, determines what kind of infection you're going to have or the pattern of infection. So that's what we're talking about, whether it's acute or persistent. It's a function of what the virus does and how the host responds. And here is a graph which I believe you have already seen, which illustrates in a graph format what we're talking about. Here on the top is an acute infection, and the information here in the blue line is the amount of virus produced with time, so virus production with time. And then the red area is the amount of disease produced in the host. So in acute infection, you have a burst of virus production accompanied by disease, and then the infection is over. It's a relatively rapid course of events. And typical acute infections are caused by rhinoviruses, common cold viruses, rotaviruses, which we will talk about today, and influenza viruses. So this is what we'll talk about today. And Dr. Silverstein, next time, will talk about these various kinds of persistent infections, which basically just go on for a long period of time. And you can have virus produced for a long time, as in here, the lymphocytic choriomeningitis virus. Eventually, there's disease and death. Herpes viruses, you have repeated bouts of production of virus, either with or without disease. And then long infections where there's initially virus and disease produced, and then a smoldering period eventually leading to death. And this is characteristic of HIV. But today we'll focus on acute infections. Again, these are relatively brief. Everything is relative, of course. But in terms of a persistent infection which lasts years, this is much shorter. In a healthy host, within days, the viruses are eliminated. And usually you get one infection, so you can make vaccines against these viruses. If there are multiple serotypes, of course, you can be reinfected, depending on how many serotypes you are. And some viruses utilize this to get around the immune response. So some infections can be overwhelming. I don't want you to get the idea that these are benign infections for the most part. They can be life-threatening, even in healthy hosts. So influenza, which is an acute infection, kills 30 to 40,000 people a year in the U.S. alone. 30 to 40,000 people. So that always amazes me when people say, I don't need a flu vaccine, because they think you may not be one of those 30 or 40,000. There are many zoonotic infections where a virus goes from an animal into a person that can also be life-threatening. It's an acute infection. It's relatively rapid, but because it's a new virus for the host, they can kill you. So here's the course of a typical acute infection. Again, you've seen this, but repetition is wonderful for establishing concepts. It's a graph with time, of course, and virus growth. You get infected here. The virus begins to multiply. The innate defenses, of course, kick in immediately, and we've talked about those extensively here. If the level of virus goes above a certain threshold that the innate defense mechanism can't deal with any longer, then adaptive responses are induced. We talked about the communication between the innate and adaptive responses that cause that trigger. Virus keeps replicating. Eventually, if you're unlucky, the adaptive response kicks in. Of course, there's a 10 to 14-day delay before you have a good adaptive response made. Virus levels will decline, and eventually the virus is cleared. So there are two outcomes with an acute infection. You clear the infection or you die. That's really it. And so if you're unlucky, the latter occurs. And then, of course, you have memory. Your immune response remembers the pathogen, the antigenic pattern of the pathogen. So if you're reinfected with a similar virus, you will be protected because the adaptive response will be much more rapid. With all infections, there is what we call an incubation period. That's the initial period before the symptoms are obvious. In that time, viral genomes are replicating. So the virus has entered you, they're replicating, and they may or may not cause symptoms that are obviously associated with the virus. You're, of course, responding within hours. The innate system has detected infection. You begin to produce cytokines. And remember, those are produced locally, but they diffuse. They diffuse all over your body, and they have global effects. Remember the fever, malaise. I don't want to get up today. I feel lousy. Aches, pains, nausea. These are all global effects caused by an acute virus infection, and that's the initial innate response. These are not typical of any particular virus infection. They're nonspecific symptoms, so you can't really tell what you have when you have these initial global effects. The incubation period can be very short, or it can be very long, and whether we actually call those virus infections acute infections where the incubation period is years is a matter of debate or not, but I'll show you a chart that illustrates that in a moment. Now, short incubation periods, you'll see within our definition of acute, and of course, like everything else in science, we apply definitions to try and categorize things so we can study them better. And so there are always examples that span our definitions. A short incubation period, it usually means that the virus infects locally and all the disease is produced by replication there. So a respiratory virus like flu replicates in your respiratory tract, and those where the symptoms are produced. So the virus doesn't have to go far to cause the symptoms, so the incubation period is short. Right? Pretty straightforward. Influenza, as an example. Long incubation periods mean that the virus needs to go somewhere else to cause disease, and it takes time to get there. And it may also be that there are host responses involved in the disease also, so the incubation period is longer. An example of this is measles. So here is a table with incubation periods of some common viral infections. These are not all acute infections, but I just want to show it to show you the range of incubation periods from 1 to 2 days for influenza, a couple of days for a common cold, these respiratory viruses. So again, you see the pattern here. The pretty short incubation periods are those infections where the symptoms are produced locally. And then you get into increasing incubation periods, enteroviruses, poliovirus, measles virus, where there's initial entry point and replication, then the virus spreads somewhere else to cause the actual disease. Remember, the nonspecific symptoms, the fever and aching, that's not what we're talking about in terms of the incubation period leading up to. Those are actually known as prodromal syndromes. They're not really specific for any virus. The incubation period, say, for polio is 20 days until you get paralysis. not just a fever or muscle aches. So you can see some of them are quite long. Then we move into persistent long-term infections like Hep B and C, the papillomaviruses, and AIDS, up to 10-year incubation. So I wouldn't consider that to be an acute infection, although you could view it as an acute infection with a very long incubation period, except that initially when you are infected with HIV-1, there is a burst of virus production and disease, and then it stops and goes on much longer. Rabies is an acute virus infection. It just takes a long time for the virus to get from the dog bite to the CNS. And the farther you're away from the CNS that you are bitten, the longer the incubation period, hence 30 to 100 days. So with rabies, you have time to immunize someone before the virus enters the CNS. There are also inapparent acute infections. Now, don't think that every acute infection gives you symptoms. There are ones that don't give you symptoms. We call those inapparent. They are successful infections, though, in the virus's terms, because they produce viruses. That's what the virus wants to do if viruses, in fact, want anything, which we know they don't. They need to produce progeny, and whether they produce disease or not may not matter. You make enough viruses in these inapparent acute infections to spread, but there's just not enough to make the typical symptoms of the virus itself. You may have a slight fever or some achiness, and then it goes away because the virus never gets to the end point. And you may not think anything of it. You'll come to class and spread the infection to others. Or you may not have any symptoms at all and you're shedding infection. So these are inapparent acute infections. How do we know that these occur? Well, many studies have been done in populations looking at antibodies against specific viruses. And you can see in a population there is no disease, yet there is a rise in antibody titer, which means that these people have been infected. Well-adapted pathogens often follow this pattern. A virus does not have to produce disease in order to spread. Of course, in some cases it helps if a virus induces coughing or sneezing, but it isn't essential. So well-adapted pathogens that don't kill their hosts have this pattern of inapparent infections. So a good example is over 90% of polio infections are inapparent. And that's why this is a tough virus to detect in the population. At least years ago when it was rampant, very hard to detect because most of the people who were infected and shedding and transmitting to others didn't have any symptoms. So you couldn't put them away until the virus was gone. So quarantining was actually used to try and prevent the spread of polio in the US, but it was absolutely useless because of this number. You had some people with paralysis, which are 1% of the total infections. They're in the hospital, they're quarantined, and the rest of the 99% are walking around with mild infections spreading the virus. Yes?
So is there some sort of evolutionary advantage to a virus that causes an inapparent infection?
Is there an evolutionary advantage? That's a good question. I'm not sure that it matters. I think it depends on the virus. For example, measles virus requires a very large population of susceptible individuals, and if it kills the host, then it's not going to be able to spread effectively. So I think it depends on the virus. This is something we talk about all the time. Is it of any value for a virus to become more virulent? And I think not in most cases. It just needs to spread. Transmissibility is what is the major factor that it really is selected for. It may be that higher virulence goes along with that accidentally, but I don't think it's needed. So I would guess that these kinds of inapparent infections are actually fine. And if we did more sampling of healthy populations, I think we would find that there are a lot of inapparent infections that we don't know about. I think in the animal kingdom, in the animals out in the wild, non-humans, I suspect there are many, many unknown virus infections, and that the animals are fine. It's a perfect example of adaptation to this kind of lifestyle. Now, acute infections are common public health problems for a number of reasons, and they are most frequently associated with serious epidemics. Most frequently, of course, AIDS also infects millions of people, but that's one virus. But in general, the acute infections are those historically that have been associated with outbreaks: polio, influenza, measles, that occur each year. Smallpox, of course, before it was eradicated. They are often associated with viruses that go from animals to humans, and these again we call zoonotic infections. Ebola viruses, is a zoonotic infection, going from a bat to a human, and there are many other examples of that as well. So these infections are hard to deal with in terms of public health. So people who deal with human health, physicians, epidemiologists, drug companies, public health officials, because by the time you feel ill or make an immune response, most acute infections are over. So if you remember the curve, the graph we showed of the immune response versus virus, the virus peaks and then the immune response is really kicking in and really the infection is over for the most part. So you feel ill, it's really kind of late to do anything about it if you have an acute infection, with a few exceptions. Virus has already spread on, so you can't stop it. You're shedding virus in the early days and you're spreading it on to the next host. So it's really a good adaptation evolutionarily. That's what these viruses have adapted to do. Replicate very quickly early on in the absence of symptoms. They're hard to diagnose, and in large populations, in crowded situations, extremely difficult to control. And that's why in certain countries, people wear face masks all the time. We don't do that here in the U.S., but I'm sure you've seen photographs of other countries, people wearing face masks to try and get around this issue. And as the world has aged and the population has increased, we have increased the number of crowding situations that foster virus spread, like daycare centers. There was a time where there were no daycare centers, right? Mothers stayed home and took care of the kids. But now we have daycare centers where all the kids exchange their virus infections and then they bring them home to their parents. And I can tell you I have firsthand experience at that. It happens all the time up to a certain age. Military boot camps, this has been around for a long time, and this is historically a good way to spread infection. College dorms, nursing homes, schools and offices, increasingly crowded, great ways to spread acute infections. And often you can see very explosive spread of particular infections under these conditions. If you read the infectious disease literature, you will see at a regular basis, for example, outbreaks of gastroenteritis in daycare centers, nursing homes, colleges, always happens in explosive outbreaks because the virus is being spread before there are obvious symptoms. Very difficult to control. So this is as an implication for antiviral drugs when they are available. They have to be used early in infection. So for an acute infection, if you want to treat these with antivirals, you have to catch the infection before symptoms are manifested. By the time the symptoms arise, it's really almost too late. Now, persistent virus infections don't have this limitation. Right? If AIDS is incubating for 10 years, you have a lot of time to interrupt with antivirals. Hepatitis C also. But acute infections that are very short, this is an issue. An example of this is Tamiflu, the inhibitor of influenza virus. You have to take this within 48 hours of getting sick. Otherwise, it's of no value whatsoever. So you're faced with a conundrum because most people, when they first feel ill with flu, are you going to rush to a clinic or a physician and then get diagnosed? There are rapid diagnosis kits, but the efficiency of those is pretty poor. And then maybe the physician will say, well, it may be influenza because it's going around here. Here's some Tamiflu. So that's the scenario. Most of us stay home, and then after 2 days, it's too late to take Tamiflu. So how do we defend against acute infections? The primary natural defense, of course, is our innate response, which is very important. The adaptive response doesn't kick in for at least a couple of days. Of course, adaptive cellular responses in particular are very important for resolving these infections, but the antibody responses for the most part don't do anything. The adaptive response clears your virus and provides memory. Now, what would happen if you had a strong innate defense but no adaptive defense? So you can actually do this experimentally in animals, and the answer is that without innate defenses, the infection can spread from the primary site of infection. So the innate response, of course, is central in controlling the infection, and if it can't control it, the adaptive response has to kick in. In people without immune responses, immunocompromised individuals, they may not have either B cells or T cells, or they may not have either. Infections, acute infections in particular, can be disastrous. Now, the memory aspect of acute infections is important. Acute infections are typically usually one-time only infections. And therefore, we can make vaccines against the agents to provide protection. And in fact, we learned that vaccines could work many years ago from memory in people. People who had an infection didn't get reinfected again. Edward Jenner noticed this in terms of smallpox, and we'll talk about that in the vaccine lecture. But there are always exceptions, and antigenic variation can bypass adaptive immune defenses. So by antigenic variation, we mean, for example, influenza virus undergoes a yearly antigenic variation. So if you get a vaccine this year against the currently circulating strains, by a year or two from now, the strain will have varied sufficiently so that the memory that you have isn't enough to prevent infection. Yeah?
How is it able to vary like on such a regular cycle of 1 year?
Well, remember, all viruses make mistakes in replicating the nucleic acid. The RNA viruses can't correct the mistakes, so they have a very high error rate. So all RNA viruses make a lot of mistakes. And some can accommodate those changes in their proteins. So influenza virus, for reasons we don't understand, the glycoproteins on the surface can undergo enormous changes. They're all localized near the receptor binding portion, and it can accommodate those. Other viruses can't. They don't do. Measles virus hardly varies at all. There are structural constraints. So that's the only answer I can give you. We don't understand the basis for that, but some viruses are better at accommodating than others.
Is it because of the vaccine that was out for that current year?
No, I don't think the vaccine really pushes it because not enough people are ever immunized in any 1 year to make a difference, probably. It's just that the population immunity pushes the virus to mutate or selects for viruses that escape the immunity. The alternative is rhinovirus. It doesn't undergo variation, but there are more than 100 different serotypes circulating at the same time. And so in any given year, you're likely to get another common cold. Yeah?
Do you know what threshold is necessary for herd immunity for flu?
For flu, I don't know offhand, but that's different for every virus, right? I don't know what the number is for flu at the moment. Let's say 100%. So you get vaccinated. Okay, so we're going to talk about some specific examples of acute infections. I picked these 5 because they're transmitted in slightly different ways. And just so you can learn about how acute infections develop, how they're transmitted, and some of the details dealing with them. So the first one we'll talk about is influenza virus. And here is an influenza virion at the upper right. If you remember, it is an enveloped virus. The RNAs are negative stranded, and they're present in segments. So these viruses can undergo reassortment, which is another way to have genetic variation. There are 3 types of influenza. They're called influenza A, B, and C. The A and B influenza strains cause a very similar respiratory disease. Influenza C virus is mostly inapparent infections. So there's a lot of transmission of influenza C that we've learned about through serosurveys, but very little disease associated with it. So we don't immunize against influenza C because it doesn't really cause significant disease. We do immunize against A and B. Influenza A strains cause pandemics. These are the outbreaks, the global epidemics, large outbreaks of influenza every X number of years, 20, 30, 40 years. We just had a pandemic in 2009. Does anyone know when the previous pandemic was before 2009 of influenza? Sooner than that. It was the year that Richard Nixon was elected president the first time. I might be wrong on that. Does anybody know that year? 68? Was he elected in 68? But it doesn't matter. It was 1968. So that was a long period. That was a long period between 68 and 2009. The one before 68 was 57, so that was much shorter. And then before 57 was 1918, so again, much longer. So it's not terribly regular, but every so many years there is a pandemic, and that only happens with influenza A virus. This virus, the hallmark of this is antigenic variation. Every year or two, the virus changes enough to evade your immunity, and every X number of years, it changes substantially, so you have a big outbreak. Yes?
What allows a virus to be able to cause or not cause a pandemic?
It's probably a number of factors. So let's say we have a strain of influenza circulating for 10 or 20 years, it changes a little bit every few years, but there's still a large population immunity against it. And then if another strain is introduced into the population, and the population has no immunity against it, that will cause a pandemic. So it's dependent on the introduction of a new strain, probably from an animal vector. So 2009, we believe that a new strain came from pigs. And it resorted in pigs, and then it got into people, and because there was no immunity, and it also had the ability to transmit effectively in people, it caused the pandemic. So pigs, we have created this reservoir because these pigs are the pig industry that we grow up, and they happen to be very susceptible to various strains of influenza. They get infected by their handlers, they get infected by birds that fly overhead and drop on them influenza viruses, so they turn out to be a very good mixing vessel. So influenza viruses are respiratory pathogens. They enter your respiratory tract by inhaling, remember, aerosols containing virus that you inhale. Someone else has produced that by coughing or sneezing or talking. Or you may touch a contaminated surface and put the virus into your nose. It was a very interesting study done about 5 or 6 years ago. It's a Swiss virologist. He took banknotes in Switzerland, and they brought them into the lab and cut them up and looked for influenza virus on them. And actually what they did is they put virus on the banknotes, and then they saw how long it lasted. And they found that it could last over a week on the banknotes. So, you know, people are going in there and taking their money out, and they're rubbing their nose and put it on the money. Someone else could get it. And in fact, they found that if there's mucus with the virus, it lasts even longer. It protects it. So you can get it by contact, not just by aerosol. So, yeah, it's only in Switzerland. Where else would you do that study, right? So the virus enters your upper tract. It can multiply there and cause a variety of these upper tract clinical manifestations, rhinitis, pharyngitis, laryngitis. But influenza is very characteristically able to go down the tract. It moves down the respiratory epithelium all the way down into the trachea. And that's why when you get flu, you really feel this infection in your chest. You can absolutely distinguish it from an upper tract infection. In the most serious cases, it gets down into your alveoli and causes pneumonia, essentially, and it can be a big problem. So you can see here, influenza virus can cause tracheitis, bronchitis, bronchiolitis, and bronchopneumonia, all the way down into the alveoli. So it can go up and down the entire tract. Uncomplicated influenza, the incubation period is short, 1 to 5 days. And depending on how much virus you ingest or inhale and how good your immune system is, all of our immune systems are different. You can pinpoint to the moment the time you get flu. You can absolutely say, right an hour ago I started feeling lousy. You get a headache, chills, dry cough. I mean, in retrospect, you pinpoint it, because when you get this, it could just be the virology exam that's causing the headache. But then as the symptoms increase, you say, that's when it began. It's followed by a high fever, muscle aches, laziness, anorexia, and you have a peak of fever within 24 hours, quite high fever, and then it begins to go down after a couple of days. So your fever goes down. It's gone by day 6. And when the fever goes down, you think, I'm getting better, but no, virus isn't done with you yet. These symptoms, by the way, bless you, these symptoms can differ in very young kids or old people because they have very different immune systems. As the fever declines, then you start to cough and sneeze more. Respiratory symptoms, you go to a productive cough, which means you have a lot of mucus. It goes from being dry to a hacking, productive, mucousy, wet cough. And this can go on for 1 to 2 weeks. And during that time, the virus is replicating throughout your tract. And this depends on the receptors. Human influenza strains can replicate throughout the entire human tract because the receptors for those are in the upper and lower tract. If you happen to be infected by an avian influenza virus strain, they only like certain receptors which are present in your lower tract as well. How do you diagnose it? It's very difficult because it's a quick infection. But we do try and diagnose it because we have antivirals, and we'd like to use this especially in people who are at risk. That's why I said before, if you say you have a nursing home and you have evidence that there's an outbreak of influenza, you just treat everybody with antivirals. You don't even waste time because it's a very at-risk population. So influenza causes what's known as ILI, an influenza-like illness. Very hard to clinically differentiate from other virus infections. High fever, these are the criteria though. High fever, cough, or sore throat. That's it. So the CDC definition of influenza-like illness, high fever, cough, or sore throat. If you have that, they say you should get Tamiflu. Of course, there shouldn't be any other known cause of illness. Now, here's rapid lab tests. What role do they have? There are some tests that can be done in the doctor's office, which just involve taking some respiratory secretion and putting it on a stick of some kind, which looks for viral antigen. It develops very quickly. They have about a 50% success rate. So they'll say, no, you don't have flu when you do have flu half the time. So they have really poor accuracy. The best tests, of course, are PCR-based tests, but if you go to a doctor's office and you're coughing and hacking, the physician's just going to prescribe Tamiflu, especially if it's flu season, and you're not going to be doing a PCR test. PCR tests will be done to study spread of the infection in a population, but on a regular basis, not likely. So here are some graphs that show you symptoms versus virus shedding and immune responses. Let's start in the upper left here. This is actually an experiment that was done in volunteers. You can infect volunteers with influenza virus strains. You have to use strains that don't kill you, of course, very, very mild strains. And then you can measure symptoms and secretion. So that's what they've done in this experiment. They inoculate these individuals. They pipette virus into their noses. And then at different times after infection, they look at virus shedding, which is the dark circles here, and the symptom score. So you have to make a qualitative judgment, coughing, amount of mucus. For some of these, they actually make you blow your nose and they weigh the tissues. And they express it in grams of snot, as we used to call it. So here you see that there's not a big difference in the amount of virus, when the virus production occurs and the onset of symptoms. You see at day 1 here, there's already shedding and there's already some symptoms, and they both go up pretty much in parallel. So that's good, because as soon as you have symptoms, it means you're starting to shed virions, but not before that. So for flu, that's a good thing. It's not the case with all acute virus infections. Here we're looking at virus production compared with interferon in the dashed lines, and then various antibodies in the nasal wash and in the serum. So you can see the virus peaks at about 2 or 3 days in this experiment, goes down rapidly, and look at the antibodies are coming up way after the virus is already gone. So in this case, in influenza, the antibody is not playing a major role in clearance. All this virus is cleared by the cellular response pretty much. And then, of course, you do have memory, which will help next time you get infected. Now, influenza is a seasonal infection. In our climate, we have a temperate climate here. It happens in the winter. And you can see it by this graph, which is looking at influenza of different types, A and B, and different subtypes, which we haven't talked about, but there are also A and B. A's are classified into different subtypes, as are B's. So you can see there's a peak here in the winter months every year, 2004, 2005, 5, 6, 6, 7, and 7, 8. Very reproducible, very little activity in between. This is just the number of samples that are positive for influenza here. You can see here 3,000 peak here, and these are done by CDC collaborating labs, and then they're subtyped as well. So it's a seasonal infection in temperate climates. In other climates, it also displays some seasonality, but happens at different times of the year. Now, what causes the symptoms of flu and the tissue damage? Well, the virus replication does. The virus is cytopathic and it kills cells, but there's also good evidence that the immune response plays a big role in the lung pathology. These are sections from lungs of mice that were infected with influenza virus. And here's a normal lung. You can tell it looks very nice. There are lots of airways. There are no infiltrating immune cells. This is a mouse infected with influenza virus. These dark cells are all immune cells that are infiltrating into the infected area. They're trying to clear the infection. But in fact, they also damage cells, because some of these are cytotoxic T lymphocytes. They're trying to kill infected cells, and in your lung it will cause lung pathology. The bottom two panels, they've treated these mice with a particular drug that inhibits certain cytokines that are produced by these lymphocytes. And you can see with this one, number 4, there's markedly less infiltrate in this mouse lung. So you can't get rid of it entirely, but you can reduce the infiltrate. In these mice, the infection is less lethal. So you dampen the cellular immune response, you decrease the lethality. So there is some evidence that, in fact, the pathology is immune-based. So it could be that at some point in the future, in certain at-risk populations, we might be using such inhibitors to prevent serious lung disease in influenza. You get some complications besides the primary infection. You can get primary viral pneumonia. As I said, the virus can go all the way down into the alveoli, and that's quite serious. You can also get secondary bacterial pneumonia. After the first infection of influenza has occurred, then the bacteria move in, and you get a bacterial pneumonia. Generalized muscle pain, known as myositis, sometimes heart involvement. The virus is not replicating there, but there's some secondary consequences of infection, perhaps having to do with respiratory difficulty, pressure in the pulmonary trunk, and so forth. And a syndrome called Reye's syndrome, which is an involvement of the liver and the central nervous system. Again, there's no virus replicating at those sites, but there is pathology associated there, and we don't really understand how the virus triggers it. What do you do about influenza? You go to the drugstore and you can buy all the over-the-counter stuff that may or may not make you feel better. Or you can get prescription antivirals. There are 3, Tamiflu and Relenza. Those are the trade names. These are the chemical names. These are inhibitors of the neuraminidase, which is one of the 2 glycoproteins on the surface of the virus particle. And we will talk about how they work in the antiviral lecture. And then there's an older drug called Remantadine, which is an inhibitor of the ion channel that's in the membrane of the virus. And we'll talk about that mechanism as well. Of course, there's always a vaccine, and we always tell people to get it every year, but the rate of vaccination in this country is not very good, and hence there's always influenza every year. Okay, that's flu. Let's go to polio now. This is a virus transmitted by a very different route, and it's a very different virus. It is a non-envelope virus. It has a shell, an icosahedral shell, made up of 4 different proteins. It's rather small, 30 nanometers in diameter. That's an electron micrograph of the particles. The structure of this virus was solved many years ago by X-ray crystallography, and this is the kind of picture you can make when you have the coordinates of every atom in the virus particle. So you can build models like this and see what they look like. So the capsid, of course, made up of 4 different proteins, VP1 through 4, repeated with icosahedral symmetry, giving you all those nice axes of symmetry, like that 5-fold axis of symmetry right there. The capsid, of course, exists to protect the viral RNA, which is inside it. These are plus-stranded RNA viruses. Pathogenesis of polio, you ingest it. You acquire infection by fecal contamination because the virus is passed in the feces, so a new host acquires it by contamination. You ingest it, it goes down into your intestinal tract. It infects the mucosa of the intestine, passes through the basement membrane, causes inflammation of the mucosa, passes through to the lymph system and then on to the blood, and it causes a viremia. From the blood, the virus can get into the CNS, and it does so very infrequently, but when it does, it causes poliomyelitis. We think it gets there either by direct penetration from capillaries in the CNS or maybe by axonal transport. So muscles, of course, are vascularized. They carry the virus to the muscle, and virus can get into the end plate of nerves in the muscle and can be carried up into the spinal cord by axonal transport. And once they're in the spinal cord, the virus replicates in motor neurons. No other cell type in the spinal cord. It kills them, probably by lysis. This is a lytic virus. And then if you have enough neurons destroyed, you get paralyzed. This is a mouse that we made susceptible to polio many years ago. Now, polio is a uniquely human infection in nature. It only infects people, nobody else. And in fact, in the lab, you can infect other animals. You can infect certain kinds of non-human primates, but no other animal. Mice, for example, cannot be infected because they don't have a receptor for the virus. But a number of years ago, one of my students cloned the gene encoding the receptor for polio that's shown here. It's a transmembrane protein. And then another student took the gene for that protein and made a transgenic mouse. And if you then take these transgenic mice, they're now expressing poliovirus receptor from humans, and infect them with polio, they develop poliomyelitis. So the receptor is the only thing between poliovirus being able to replicate in a mouse. So that's nice because it gave us a nice model to study the pathogenesis of the disease rather than having to use much larger animals. And so you can do experiments like this where you infect the mouse with poliovirus, and then when the mouse gets paralyzed, you take the spinal cord out and you do sections, and you ask, where's the virus replicating? So here, this is a mouse that had been paralyzed when the section was taken. You can see all of these immune cells infiltrating into the area. They've come into the spinal cord to try and clear the infection. They're brought there by cytokines that are produced by the infected cells. So lots of cells of, we don't know what these are. We never actually looked, but they're probably lymphocytes and macrophages of various sorts. And then the green dots are where the virus is replicating. We hybridized this section with a RNA probe that would detect virus RNA. And these are neurons. You can tell they're large cells. They're triangular shaped, pyramidal shaped. And those are the neurons that the virus is replicating in. And again, if you destroy enough neurons that are innervating a particular muscle, the muscle becomes paralyzed. So that's the basis of the paralytic disease. So here's a nice depiction of the time course of polio. This is taken from a textbook from the 1950s. So medical textbooks don't have polio in them anymore because we don't have it in this country. But in the 50s, there was plenty of polio, so every textbook of medicine had chapters on poliomyelitis. So this is a hand-drawn figure from that time. And what we're looking at here is the time course of polio and various things that are going on. So here's the time up here, the different phases of infection. We're looking at temperature here, so you can see. So here's exposure to the virus. After about a 7-day incubation period, we have a spike of temperature, headache, sore throat, nausea. Nonspecific symptoms of a virus infection caused by cytokine production. Most of the people get well, and they go on and they're fine. But 1% of those individuals, the virus then remains in them. You get another spike in temperature accompanied by stiffness, pain, and paralysis between day 10 and 15. So 1% of infections result in invasion of the CNS, and you get the paralytic disease. And look at the virus production. So here's virus in the feces. So it peaks at about the first time of temperature here, after day 7, and then it continues for a long period of time. So you excrete virus in the feces for over a month, whether you have paralysis or not. So that's how you spread the virus to others. Here is another graph of virus in the blood and antibody, just to show you that here's the initial peak of virus that coincides with that first bout of nonspecific symptoms. And then it goes down on its own, and the antibody comes up much later. So the antibody is not responsible for clearing that virus. And then virus in the central nervous system is shown on the bottom. Just after day 10, virus goes up in the CNS, and it peaks at the time of paralysis. So this only happens in individuals who have paralytic symptoms. Yeah?
How do they get people to try this out on where they are?
Now, these are people who had polio. In the 50s, you had thousands of people with polio in the U.S., so you could take specimens. You don't give them polio, but they got infected, and you could take serum, and you could take CSF to look for virus in the CNS. So all of these things, you can sample feces, serum, you take the temperature. So these are natural infections. You can't do this anymore because we don't have it. Yes?
How do they estimate, like, when the incubation time, like when they were first infected, these were actual people suffering?
So sometimes in family situations, when one child had polio, you could roughly estimate when the others were infected because they would be brought in to the hospital sometime later, and you could do a history and find out when the other child was infected. So a nice summary of the whole time course of polio. The pathogenesis, as I said, humans are the only known reservoir. It's spread by fecal-oral transmission. It's also a seasonal infection in temperate climates. It peaks during the warm months We're not sure why, but probably has to do with children being outside and playing with one another during this time. There is a complication. If you have paralytic polio, you may get something called post-polio syndrome, which happens 30 or 40 years after the initial paralysis. And I should say that many individuals can be rehabilitated from having polio paralysis. You can have your limb function restored by therapy. Sometimes the virus does replicate in respiratory control centers, and then you have respiratory failure. That would get you in an iron lung, but even then you can eventually come out of it. So not a high fatality rate, but a lot of these individuals who were having normal lives 30 to 40 years later would get paralyzed again. So that's called post-polio syndrome. About 25 to 40% of the original paralytic cases this occurred in. Doesn't seem to be any virus present in these individuals. We're not sure what's happening. One of the theories is that normally as you age, you lose so many neurons at a certain rate. And if you've lost many to polio, then you have fewer backup neurons. So then as you age, you accrue another paralysis. But that's a theory. There's really no proof for it. Of course, now that there's no polio in the U.S., there's less and less post-polio syndrome in the U.S. This is what has happened with polio in the U.S. from 1950 to 2007. Between 15 and 20,000 cases a year. These are paralytic cases. So remember, 15 to 20,000 paralytic cases. The paralytic rate is 1 in 100. So there are many more infections going on here. The inactivated polio vaccine was introduced in 1952, and it eliminated most of the disease in the U.S. The live infectious vaccine in 1962, and the last case of polio in the U.S. 1979, indigenous meeting caused by wild-type virus. And we'll talk about this more in the vaccines lecture. But right now, there's no more polio. It's considered eradicated from the U.S. and many other countries as well. Next virus is Measles virus. This is a paramyxovirus. It is one of the most contagious human viruses, highly infectious. Before vaccine, 40 million infections per year, 250,000 deaths per year, mostly in children. And it requires a substantial population for transmission, between 300 and 500,000 individuals. So small isolated populations don't have measles because they can't sustain the transmission of the virus. The virus is an enveloped RNA virus. The genome is negative stranded RNA. It's present in a helical configuration wrapped up in a variety of different proteins, including, of course, the RNA polymerase. This is a negative strand virus, so it has to bring in a polymerase with it. There is an envelope around the genome, and in the envelope are the glycoproteins, the hemagglutinin and the fusion protein, which are involved in attachment and fusion. Here's an electron micrograph of the particles. They are larger than polio and influenza viruses, slightly large viruses. Pathogenesis. There is one serotype of measles virus, and as I said, there's really very little antigenic variation. So once you get infected, you're pretty much protected for life. It's transmitted by respiratory secretions. So you sneeze or cough, you exhale virus in droplets, and you infect others. Here now we have a different situation from polio. There is a period of shedding of virus 2 to 3 days before the rash occurs. So when measles was rampant in the U.S., it was quite clear when kids had measles because they were covered with a rash. But before that, they were shedding virus, and that's why it spread so effectively in schools and other settings, because the children weren't kept at home. Nearly all infected individuals show signs of disease. Again, very different from polio, that only 1% got paralytic disease. Here, almost 100% of people infected with measles will show the rash disease. Pathogenesis, you inhale the virus in an aerosol. It multiplies in your respiratory epithelium and then enters the bloodstream. So it's not like influenza, where it remains in the respiratory tract. This virus enters the bloodstream and then spreads to other organs. So it has a longer incubation period as a consequence. There is then a secondary viremia. Remember, the virus multiplies in these organs initially, then there's a secondary viremia, and that brings virus to the skin, where it causes a rash. And the rash is a delayed type hypersensitivity reaction. It is virus multiplying in cells and T cells coming in to try and clear the infection. They make the rash. There are also rare complications of measles that we'll talk briefly about. It can enter your brain and cause encephalitis, or it can cause a very interesting disease, subacute sclerosing panencephalitis, SSPE. And this is something that happens many years after you get measles. So a child will get measles, they recover, they're fine. 30 years later, they start to have this progressive neurodegeneration. They eventually die. It's 100% fatal, and it's caused by the initial measles infection. So here's a timeline of some of these events. You have infection here. There's a primary viremia. The virus spreads to other tissues. So that's the incubation period. Then there's what we call a prodromal period, which is the period just before the rash. So the rash is occurring at around day 14. So it's a 2-week incubation period. And you can see that that's plenty of time for these kids to shed virus and pass it on to other people. At the onset of rash, you have antibody produced and then eventually recovery. So uncomplicated measles is associated with a high fever, cough, and symptoms of a cold, runny nose, conjunctivitis, and Koplik spots. These are white spots in the mouth, which are shown on the bottom here. I'm not sure you'll be able to see this. But in the day when measles was rampant, this was diagnostic for measles. If the kid had these white spots, they're right down here, tiny white spots on the mucosa inside the cheek, it was pretty much clear that was measles. The rash is all over the body, face, extremities, trunk. So this is the delayed type hypersensitivity rash caused by virus infection. This virus, too, has complications. Encephalitis, as I said, the virus can get into your CNS. 1 in 3,000 cases. So 3,000 cases of measles, one of those encephalitis. SSPE many years later. And then, as we talked about a few lectures ago, this virus causes immunosuppression. So as a consequence, you don't have a good immune response against other pathogens that you might encounter. So you get secondary infections as a consequence of having measles. And that's the main cause of death in third world children who get measles. They get immunosuppressed and they get other infections, parasites or bacteria. So how do you prevent measles? Well, in the U.S., before a vaccine, there were 3 to 4 million cases a year, 400 to 500 deaths, and 48,000 hospitalizations. And interestingly, or importantly, 1,000 cases of chronic disability caused by encephalitis, the virus multiplying in your brain. So viruses in your brain is not a good thing. They can wreak havoc, and one of them is this. We stopped transmission of measles in the U.S. by 2000 by using the vaccine, that is endemic transmission. There are still populations in the U.S. who refuse to be vaccinated, so there are outbreaks of measles all over as a consequence. So right now, the vaccine we use is composed of 3 different viruses, measles, along with mumps and rubella viruses, MMR. Back in 1998, some of you may remember, a British gastroenterologist named Andrew Wakefield claimed that the MMR vaccine caused autism. This study has since been refuted over and over, but still parents refuse to immunize their kids because of this. And there are always outbreaks of measles. There have been quite a few in the UK and Ireland, and then people traveling bring them to the US, and they cause outbreaks here in the US in under-immunized populations. So we don't have measles circulating, but it can be brought in from Europe where there are large outbreaks. So here's a graph showing you the impact of the vaccine. Here are cases of measles per year in the US. 600, 800,000. The vaccine licensed in the 60s, and you can see it eliminated measles. Now we don't have any more circulation of measles virus. So this is measles disease, and this is measles-induced death. So the vaccine is quite effective. Okay, the next virus, the next acute infection, gastroenteritis. So in 24 hours, anywhere in the world, about 200 million people have gastroenteritis. Gastroenteritis is an inflammation of the intestinal tract. accompanied by diarrhea. And the amount of diarrhea these individuals pass, these 200 million people, equals the volume of water falling over Victoria Falls in 1 minute. That is a lot of water. 65 million liters per minute of diarrhea. So most of that is viral, in fact. And a big cause is rotavirus, the single most important cause of diarrheal illness in infants and children worldwide. It accounts for 30 to 50% of all severe diarrheal disease in that age group. These viruses were just discovered in the 1970s. We didn't know what was causing gastroenteritis. We thought it was mainly bacteria, and certainly some bacteria do cause gastroenteritis, but someone at the NIH was looking at mucosal biopsies from kids with gastroenteritis, and he saw virus particles, and those were rotaviruses. It turns out there are 7 different serogroups of these viruses, so you have to keep that in consideration when you make a vaccine because they're all circulating in different places. They infect many different animals besides us. These viruses are like reoviruses. They have a segmented, double-stranded RNA genome, which means they can readily reassort. So rotaviruses infect everybody. You are all seropositive for rotavirus, I'm sure, unless you're living in a cave for all your life. The infections are also seasonal. They peak in the wintertime. And as I said, they're the most common cause of childhood gastroenteritis. 25 million physician visits per year, 2 million hospitalizations, 800,000 deaths. So this is a significant pathogen. In the U.S., before we introduced the vaccine, every child was infected by 5 years of age, and 1 in 72 of those children were hospitalized. And in the developing world, in the U.S., this can be handled, although it was fatal in cases where you couldn't hydrate the child quickly enough. So the key to saving an infant that has viral gastroenteritis is to make sure you give them fluids. And in many countries in the developing world, they can't do this, and so the children die. So it accounts for 5% of all the mortality in children less than 5 years old. And this is the global distribution of rotavirus deaths just recently. So you see there's no more in the developed countries in the U.S. There's very little in Europe, in Russia, Australia, but Asia, Africa, and even South America. Each dot, I think, is 500 deaths. So you can see there's a huge burden of infant mortality caused by this, and it's really mainly because they cannot get these infants intravenous fluids rapidly enough. So this is what the virus looks like. It's a typical reovirus. It has an icosahedral shell. In fact, it has 2 of them. It has 2 icosahedral shells, one inside and one outside. And there are viral glycoproteins in this shell. They're called VP4, and then the shell itself is VP7. And I point that out because those are 2 of the more important proteins to make antibodies against if you want to be protected against these viruses. Inside is the double-stranded RNA genome. When these viruses were first seen in those biopsies of intestinal mucosa, this is what they looked like. And the guy who discovered them said, oh, they look like little wheels, and that's why he called them rotaviruses. So you can see here, they're very symmetrical. Pathogenesis, it's transmitted by fecal-oral contamination. You shed the virus in your feces, and someone else picks it up. You don't need very much virus to infect you. 10 virus particles are enough to cause gastroenteritis. And as I said, young infants are at risk for dehydration. You do get infected over and over again as you age, but the subsequent infections are milder, and eventually you don't get disease because you have antibodies that build up against these viruses. Now, you can be infected and you can shed virus even though you don't have symptoms, and so you become a carrier of rotavirus, and that's one of the main ways that it's spread in larger situations. You shed a lot of virus in the feces, 10 to the 8th to 10 to the 10th particles per milliliter. So just think of all that 65 million gallons, is that liters, per minute, and multiply it times 10 to the 8th to 10 to the 10th particles. A lot of virus there. How do you get it? Contaminated hands and foods. You have a food handler who has an infection with no symptoms. If he or she had symptoms, they should stay home. But they have no symptoms. They're shedding virus because they've had many infections over the years, so it's asymptomatic. And they have virus on their hands. and they contaminate the food. You know, all the signs in the restrooms, restrooms of restaurants, employees must wash their hands. The motivation was in part rotavirus. There are other viruses that cause gastroenteritis as well, because we know a lot of these individuals are asymptomatic shedders, and so if they wash their hands, you can really cut down on the burden of infection. These viruses are also quite stable, so they will last on environmental surfaces, and that's another way you can pick them up. The disease is self-limiting. Vomiting, watery diarrhea, low-grade fever, wonderful infection to have. As I said, asymptomatic infections are important. 1 to 3 day incubation period. Then vomiting followed by 4 to 8 days of diarrhea. Sound good? But then you get better for the most part. Recovery is usually complete unless you don't take fluids. I got an email last week from someone. She said, my husband is vomiting and has diarrhea and he doesn't know what to do. She said, I think he has that rotavirus. And I said, he might, but just have him drink a lot of water. She said, should I go to the doctor? I said, no, there's nothing to do. There's no antiviral. Just have to drink a lot of water and you'll be fine. So how does it work? You ingest contaminated food or water. The virus goes into your intestinal tract. It multiplies in the mucosal surfaces there. And you can see this is a section of intestinal villi. And these dark spots are where the virus is multiplying. And that causes a fluid imbalance. In fact, a couple of lectures ago, we talked about how this is one of those viruses that makes a toxin. It makes a toxin. It makes a protein called VP4. And VP4 probably causes the diarrhea. So here's a rotavirus binding to intestinal epithelial cells. The things I want to point out here are that the infected cells produce cytokines that attract T cells, and of course those cause intestinal cell damage as they try to eliminate the infection. In this cell here, you can see NSP4 is being produced. That's the toxin produced by the virus. It causes a excretion of chloride, and that leads to a fluid imbalance, giving you diarrhea. There's also some evidence that the virus interacts in some way with the enteric nervous system. So this question mark here might be the virus protein NSP4. It could be that NSP4 interacting with this nervous system also has a role in diarrhea as well. Now there are a couple of vaccines against rotavirus because they're significant pathogens. The first one was called RotaShield, and it was a reassortant between human and rhesus monkey rotaviruses. It was licensed in 1998. So they start with a rhesus rotavirus, and these viruses don't cause disease in people. So you make reassortants of those rhesus viruses with human viruses, and so you then select for viruses that have the human glycoproteins on the surface of the particle, and all the other genes are from the rhesus rotavirus. So those viruses you can ingest. They will make antibodies against the human viral proteins, but you don't get sick. Okay? And these are just some of the reassortants that are used. Unfortunately, this was suspended a year after use because there was some evidence that this vaccine was causing in kids. So you give it to very young kids. It was causing intussusception. So intussusception is when your small intestine telescopes into the colon. All right. So it's a natural complication of a rotavirus infection. And there was some statistical evidence that it was increasing in these individuals getting this vaccine. So they pulled it off the market. It's not clear that, in fact, the vaccine was at fault here at all. It's a big debate going on about this. And in fact, this vaccine is being retested in other areas of the world. So right now, then a number of years later, 2 other vaccines were licensed, and we're using these now. One of them is called Rotatech. It's a reassortant between human and bovine strains, same logic as the rhesus human reassortants from the previous vaccine. So that's the Rotatech right here. There are 1, 2, 3, 4, 5 different reassortants. And this is so you cover all the different important serogroups. And then there's another one called Rotarix. This is actually a human isolate that was attenuated by passage in different hosts. And we talk about vaccines, we'll talk about how that works. And there's just one component in this vaccine. So these are orally administered. That's why they have the cute little teaspoons here, I guess. You drink it, multiplies in your intestinal tract, and affords protection. So these are very effective, about 80% protection against disease. Now, last year when this course was going on, a paper came out. Someone had taken these two vaccines and they sequenced it, and they found a contaminant, another virus that wasn't supposed to be there. It was a porcine circovirus. So both of these were immediately withdrawn from the market for about 2 months until it was decided that this circovirus wasn't a problem. But the circovirus came from the trypsin that is used to split the cells that are used to grow the virus vaccine. So if you've ever done cell culture, you know you grow cells in dishes, you split them with trypsin. The trypsin comes from pig pancreas, and the pigs have circoviruses. So that's where the virus came from. All right, the last one we'll talk about is West Nile virus. This one is a flavivirus isolated in 1937 from the West Nile district of Uganda. So many people, when they hear about West Nile, they think it came from Egypt, but it didn't. It came from Uganda. Flaviviruses are enveloped positive strand RNA viruses. 1937, not present in the Western Hemisphere until 1999. And that year, it arrived in New York City. Anybody remember that? It came to New York City. It was an outbreak in Queens, and it just spread all over the U.S. Somehow it got into the U.S. that summer. It may have been imported from Israel, is one of the theories, because the sequence of this virus that came to New York is exactly the same as an isolate from a goose farm in Israel. So somehow it got over here. And it spread all over the U.S., Canada, Mexico. This virus has a quite large host range. It infects hundreds of different birds, mosquitoes, and 18 other vertebrates in addition. The virus is an enveloped virus. It has a core, though, that has icosahedral symmetry. And the symmetry of the core is passed on to the enveloped glycoproteins. So if you look at this enveloped virus, it structurally has, the glycoproteins have icosahedral symmetry because of the underlying core. And here's an electron micrograph of the virus. Transmission cycle mainly involves mosquitoes. The mosquitoes transmit the virus back and forth to birds of different species. So the mosquito bites a bird, picks up virus, infects another bird, and so forth. There are dead-end hosts, such as horses and humans. So we get infected by the mosquitoes, but we don't transmit it to others. There's no evidence, at least, that we are transmitting it to any other host. So in nature, this exists in the transmission cycle between birds and mosquitoes. Dead-end hosts are horses and humans. So it's transmitted to us by a mosquito bite. The incubation period is 3 to 14 days. About 20 to 30% of infected people develop a flu-like illness. It's called West Nile fever. And 80% of those infected have no symptoms at all. So a lot of people can be infected, but this is not a consequence for spreading the virus to others because we don't spread it to other people. 150 people develop neuroinvasive disease. The virus gets into your CNS. You have headache, ocular manifestations, muscle weakness, cognitive impairment, some flaccid paralysis, and this is associated with 10% mortality. But more importantly, over half of these individuals that have neuroinvasive disease have long-term neurological sequelae, very much like the kids infected with measles where it gets into their brains. So again, not a good thing to have virus in your brain. If you remember, we talked about how the innate response allows West Nile to get into your brain. The TNF produced makes the capillaries porous so the virus can get in. Yes?
How do birds spread West Nile to each other?
That's a bit controversial. Probably grooming or mating or something like that. Yeah. So here's a little scheme of pathogenesis. The virus is taken from birds. The virus bites us. Remember, when a mosquito bites you, it deposits the virus below the outer layer of skin. And interestingly, what happens when the virus gets there, starts to multiply, and what comes in? Dendritic cells, right? Because they sense the infection. Virus infects those dendritic cells. Very smart. And then the dendritic cells go to the lymph node, and then the virus gets released, and they infect other cells in the lymph node, so they've had a perfect ride to where they like to replicate. And from the lymph node, they get into the blood. They infect other organs, like the spleen. And as we said earlier, TNF-alpha plays a role of getting the virus across the blood-brain barrier where it replicates in neurons, and that's a cause of the neurological symptoms. In the CNS of most individuals, interferon, chemokines, and a variety of T cells keep the infection in check. But for some reason, in certain individuals, they don't work properly, and you have replication in neurons and these sequelae. So this is the graph of West Nile in the USA from 1999 to 2008. The black bars are total cases, and the open bars are the CNS involvements, neuroinvasiveness. So you see just thousands of cases a year, 4,000 in 2002, a very high year in 2003, 10,000. This has to do with climate largely, because when it's very dry, the mosquitoes prefer to bite humans rather than birds. And Dixon Despommier will tell you all about this on the second to the last lecture of this course. And you can see different numbers here in subsequent years. If you'd like to know which state the virus can be found in, it's basically everywhere, although some states have more than others. 2008 on top here, you can see low numbers for most states, but California had quite a few cases, 445. 2007, overall more numbers throughout, but every state essentially having infections. How do you prevent West Nile? At the moment, there are a couple of vaccines in development because it does cause significant disease in people where the virus goes into their brains. So there are 3 or 4 different candidates, and I suspect in the next 5 or 10 years those will be licensed. I don't know who will receive them. I suppose it will be a voluntary vaccine. You decide for yourself if you're worried about West Nile. You can get it in any state, as you can see, even in New York State, but 46 cases in 2008, 22 in 2007, the risk is rather low. So I guess if you move to California, you should probably get the vaccine when it becomes available. For now, the prevention involves mosquito control, eliminating standing water. It does a lot to getting rid of the vector that spreads West Nile. So there are many, many things you can do, and again, Dixon Despommier will tell you about this when he talks about West Nile. Okay.